Hello, everyone. Welcome to Journal Club. Are you as excited as me? Of course you are. Everybody's here? We're good? Okay. We are going to be covering a paper in gene therapy today. It was published in 2020. It's called Transposon-Mediated Generation of CAR T Cells Shows Efficient Anti-B Cell Leukemia Response After Ex Vivo Expansion. That's a lot of jargon, but it actually is it's a very specific title. It's actually a pretty good title. I mean, that's a pretty good title because it's a sentence, right? So transposons, what are those? What's a CAR T cell? We know what efficient means. We know hopefully what a B cell is because it's involved in immunity. Leukemia is when those cells are cancerous. And then ex vivo means they took it out of somebody and they expanded it rather than making it in vitro, which is like homemade. Okay, so Chineme Ewenike is, did I pronounce it right? Yeah?
Ewenike.
I screwed up.
It's close enough. It's fine.
Okay. Is going to be presenting this, and take it away.
All right. Let me share my screen. All right. So let's just do some introductory stuff about the paper. So T cells, which is kind of what's going to be discussed this whole time, are just white blood cells that attack specific antigens and diseased cells. And T cells have specific protein receptors on its surface that latches on to those antigens, which allows the T cell to release toxic chemicals in order to destroy the tumor cells. And so in this paper, we're talking about CAR T cell therapy, which is when T cells are extracted from a patient and then are genetically engineered to express anti CD19 chimeric antigen receptors, or CARs, is what's basically called. And CD19 is a type of molecule that exists on the surface of cancer cells. And so these T cells are being engineered to express anti CD19 receptors in order to combat those tumor cells. And currently, viral vectors are being used to genetically modify the T cells in order to exhibit those CARs. And viral vectors are modified viruses that are genetically engineered to deliver genetic information. And then the newly modified T cells with the CAR receptors are then multiplied a bunch of times and reinfused back into the patient. And then the CARs can latch onto the CD19 molecule in order to destroy the tumor cells. And currently, this type of therapy is used for patients with CD19 positive B cell leukemia or diffuse large B cell lymphoma. And it's very expensive, and it can take up to 15 days to generate all of those cells to reinfuse back into the patient. And so in this study, the scientists are using a protocol based on the non-viral transposon vector known as Sleeping Beauty in order to generate 19 BBZ CAR T cells. And 19 BBZ is just an anti-CD19 receptor. And it's composed solely of DNA, which makes it cost much lower than viral vectors. And it can simplify the whole process of CAR T cell therapy. And in figure 1, this is kind of focusing on the first portion of the study. And the scientists are using peripheral blood mononuclear cells, which are known as PBMCs, and they're blood cells that contain various frequencies of lymphocytes. And the PBMCs were taken from a bunch of healthy donors, and then they were used to conduct electroporation, which is the process of using an electric pulse to infect cells with DNA. And so in the study, there was one group of PBMCs that were used as a mock group, and they were electroporated without plasmids or any DNA molecules, while the second group was electroporated with plasmids encoding for 19-BBZ and the Sleeping Beauty transposon. And then they were activated using a reagent that is used to stimulate and multiply those kind of cells. And then they were expanded for 8 days. And some of the results are seen here in figure 1. And for part A, it's basically looking at, on the x-axis, the mock group and the 19 BBZ group on both days 1 and day 8. And it just shows that, specifically for figure 1A, that after 8 days of expansion, that T cells were a lot more T cells were produced. And that can also be seen in figure B, when it just shows that the percentage of CAR positive cells increased during the period from day 1 to day 8. And then in part C, it just kind of expands on figure 1B, which just shows the increase in the total amount of CAR positive cell numbers. And D, E, and F are kind of showing a positive trend of the 19 BBZ cells against the mock cells. And does anybody see a problem with figures D through F?
She has thrown down the gauntlet and offered a challenge to one of you. Who's going to take the challenge? Anybody see a problem with D? I told her that the AP students, especially, should see a problem with D.
Is it the D1?
I mean panel D, sorry. I'll walk you through it. What are the asterisks? When you guys wrote your lab report, some of you used asterisks to mean something. What did it mean? Oh, it's going to be a long night. Okay, so part of doing Journal Club is that if you don't understand something and somebody points something out, you can quickly, you can call this cheating. It's really not. This is how we do it. But you can cheat and go look at the figure legend. What do the asterisks mean? It says it in the figure legend, in the paper that you read. Because nobody would come to Journal Club having not read the paper. What do the asterisks mean? People are killing Chin's rhythm here. I thought this would be easy. Do you guys not have the paper? Y'all have the paper. Go look at the figure legend. It's in the very last sentence of the figure legend. We're going to wait because this is the point of doing Journal Club, is to learn things.
It shows that significantly different.
Okay, and how did they test for significantly different data? It's that same sentence.
By finding the p-value?
By doing what test, though?
Multiple comparisons.
By using what test?
ANOVA.
An ANOVA. You guys remember ANOVAs?
Yeah.
Yeah. Question. did you get to run an ANOVA for your lab report? Did you get to run an ANOVA in
No.
your lab report? Why? I'm a terrible teacher. Wow. Why didn't you get to run an
To
ANOVA?
make sure the values weren't significantly different, or were. I mean,
No, you didn't get to run. We wanted to run an ANOVA, but we couldn't. Why?
because they were significantly different.
What was significantly different? Something was. What was?
See,
Do you guys remember the rules about statistics?
Wasn't it the p-value?
And you're skipping ahead. What are the rules about statistics? Parametric tests versus non-parametric tests. You get to run one or the other depending on what rules. Colin, I think we were talking about this earlier on the non-soccer field.
Yeah, I'm looking back in the handout on Schoology.
Mm-hmm.
Is it there with all the different tests?
No, I don't think it's in that you're talking about the table.
Yeah.
It's not in the table. I told you guys what the rules were. There were 2 rules about running parametric tests. In order to run a parametric test like an ANOVA, rule number 1 is, anybody know? The data must be normal. Rule number 2, anybody, now that I've prompted you with rule number 1, what's rule number 2?
test if the data is normal. But how? I don't know how to do that.
No, that was rule number 1. That's
Wasn't
rule
it
number,
if they were significant or insignificant?
what is?
The p-value.
No. No, you
Isn't it
skipping
testing the null hypothesis?
For what? So you guys are focusing too much on the statistical analysis itself. I'm focused on whether it was appropriate to run the test. Rule number 1 of running an ANOVA, which is a parametric test, is the data must be normal. What's rule number 2?
The group variance can't be significantly different.
Correct. Now, look at panel D. Do you see a problem?
They're significantly different.
Now, you can't really say that, but by eye, the
You can
blue
see an outlier, right?
well, the blue stuff is really spread out compared to the red stuff, which is all the same, right? Clearly, the variance is significant. Well, we don't know significantly. We didn't test it. That the test for that is called a Levine's test, in which I explained in the, when we originally did it. But clearly, by eye, those variances are not the same. You've got all the red ones in D. We're looking at panel D. All the red ones are on one little line, but all the blue ones are all spread out. If the variances are not equal, if they are significantly different from each other, you cannot run an ANOVA. They ran an ANOVA when they shouldn't. They're not allowed to. That is inappropriate. Remember, if I had the purpose of a where's my pen? There it is. The purpose of a parametric test is to compare means, right? So if I have a graph, okay, like that, the line on the normal distribution is the mean, right? But what if the graph looked like this? This one. Come on, focus. What the heck? Why are you struggling? Hold on. I don't know why it's not focusing. It's driving me crazy. But you can
Is it a double
sort of
bell?
see it, right? Well, it's a bimodal curve, right?
Yeah.
So what's the mean of this thing? It's this right here. Why isn't it focusing? This is driving me crazy. There. Okay. Bimodal curve, which means that the mean is where I put the arrow. Okay. Does that mean represent the data really well?
No.
No.
It does not. It is not. That is a non-parametric data set. You can't use means to represent this data set. You have to use something else, like medians. Okay. And which is what they do in A. That's what they did in A. Why did they switch? Right? Like, this may not bother you at all because you're not a statistician or whatever, but it means that it was not done right. Do you see? You broke the rule. Now, I said to Chin earlier, I was like, I think had they run the non-parametric test, Kruskal-Wallis, it still would have been significant. Like, they still would have gotten the result that they wanted, but they didn't do the test right. But wait, there's more. Okay, go ahead, Chin.
Should I keep talking about?
Yeah, no, keep going with whatever you got.
Okay. Well, in figure F, it's kind of showing, if you focus on just the white part, which is the CD19, it's showing that for the 19 BBZ cells that it wasn't present in either day 1 or day 8, which kind of shows that, the experiment is working. And that's all for figure 1. Should I move on right now? Is it okay to move on to figure 2? Is everybody okay with that?
Yep.
Okay. So for figure 2, it's focusing on the evaluation of T cell memory phenotype. And for the x-axis, it's showing the mock groups and the 19 BBZ groups on both day 1 and day 8. And in all, for both of them, it just shows that regarding memory, the 19 BBZ CARs induce a higher frequency of cells with a central memory phenotype, which is the white part of the graphs, which that basically means that it's a quality that allows cells to remain long-term after the infection or cancer has been eliminated, and it will provide a rapid response to past infection if it flares up again. And that would be the case for both C8 cells as seen in figure A and C4 cells as seen in figure B.
Sorry to cut you off, Chineme,
You're
but
good.
so before we started at the beginning of the presentation, you said that there were this type of cell that takes 16 days to
Oh yeah, that's what they're currently in CAR T cell therapy. They're using non-viral stuff, which is why it takes long.
Why is that?
Why does it take that long?
Yeah.
I'm not really sure. I just know that the way that it's being done in this study, which is using a non-viral way, is a lot faster. I don't know why it's that slow.
I think I do. This is not my area of expertise, but I think the difference. So you notice, the mock thing is their control, which is good. They ran a control. That's important. And it doesn't have this electroporated advantage. When Chin was describing the electroporation, you can really think of it like the paddles used in television shows to stop somebody's heart, right? Clear, right? And giving them electric shock. It works that way for cells too. When you electrically shock a cell, their membrane, because of sodium channels and a bunch of other things we've discussed in class, they all of a sudden become really permeable so that the DNA can go in. Okay, they electroporate in the mock ones. They electroporate like nonsense. It doesn't actually mean anything. So it's a good control. It's a negative control. And if you let those cells divide on their own, you'll see that if you look at how much of the bar in the second bar, how much of it's white? A lot, but not nearly as much as the ones that were transfected, right? So what happens regularly is they're dividing these cells, and they have to wait until they have enough of them to actually inject usefully into a patient. With this method that she's talking about, they're hoping that they can increase the relative number of them that are available so that they are dividing rather than the gray and the black barred cell, the ones that are listed, shown in gray and black, because those aren't the ones that are useful. It's the white ones that are useful, the white white bar. That makes sense? So it's faster. If you have more of them to start, you can have more of them to inject later. I think that's what's happening. I'm not super certain. Cool.
Okay. So in this figure, it's talking about the expansion-induced upregulation of activation/exhaustion markers. It might be kind of hard to see, but on the x-axis, it's basically the same thing that we've been talking about before. But what both of these are showing is that, on the y-axis, for all four of them, they're just genes that are upregulated by stress, I'm pretty sure. And so what these both are showing is that for the 19BBZ cells, that more of the cells are being upregulated by stress, which means that they're working a lot more with killing all the tumor cells off. So that's a good way to show that the method that's being used in the experiment is effective. Okay. So for figure 4, in this, for figure 4, I think I might have done the wrong part. Okay, no, sorry.
You're fine.
In figure 4, in this part of the study, they're using immunodeficient mice, and they're injecting them with 2 different types of leukemia cells. And one of them is known as NALM6, and the other is known as RS411. So that's kind of what you're seeing as the title of some of these figures in this part. And for figures A and B, it's just showing cytotoxic activity in the 19BBZ cells, which is the blue part. So that's just showing that they're killing a lot more tumor cells. So that's a good thing. And I guess we could skip C and D because they're basically saying the same thing. And for E, if we're focusing on the black part of figure E, that is tumor necrosis factor, which is just used for cell signaling to alert other cells in the immune system of an infection. And so for the 19BBZ, it's becoming apparent, which is a good thing. It means that they're killing off a lot more tumor cells. And were you about to say something?
Only when you're done. When you're done with the figure.
I mean, and figure F, we could skip because I think there's something wrong with it.
Yeah. Figure E, tumor necrosis factor. This is a useless piece of information, people, but the drug Humira, which is used for osteoarthritis and rheumatoid arthritis and stuff like that, Crohn's disease. The thing that it targets, it's an antibody against tumor necrosis factor. Tumor necrosis factor is a thing that helps fight tumors. So Humira is a drug that fights the thing that fights tumors, which means that people who take the drug are more likely to get cancer and more likely to also get sick in like a lack of immune reactions. So if you were wondering why I kept saying I'm immunocompromised this whole past year, it's because of that, because I'm on Humira. Okay. And TNF is important. Cool. I just wanted to make that real so you can feel bad for me. Victim points, Go ahead.
Oh, yeah. Figure F, we can just
skip it.
Okay. And figure 5 is just going off of what was in figure 4. And figure 5A is a timeline of the whole experiment from when they started multiplying the T cells to when they ended it. We can skip figure B, it's not that relevant. And in figure C, we can see that the mice that were treated with 19BBZ T cells, they showed a decreased frequency of like those like tumor cells, I think, in the blood when compared with the mock trial group, indicating that the infused lymphocytes were able to prevent leukemia progression. And in figure D, it's just showing the survival rate. And so the blue would be the highest dose, the highest number of cells for 19BBZ, and that survived the longest. So an increased, just the anti-tumor methods that were being used during the study, it showed an increased overall survival of mice being treated with 19BBZ when compared with mock T cells, which kind of just proves our whole experiment. And figure 6A is just another timeline of the experiment. We can skip figure B, and Dr. Char, do you want to explain figure 6C?
Yeah, so figure C is pretty cool because they have these special cameras that they can put in and actually observe anesthetized mice. And these mice will glow in the dark.
What does that mean? Anesthetized.
Anesthetized means they're knocked out. They don't.
Okay.
They're
anesthesia. Okay.
Anesthesia, yeah. So they're anesthetized so that you can put them into this little chamber. It's like think of it as like a CAT scan or an MRI for mice, because we actually have those. But this is actually even simpler, because these mice will glow in the dark. Okay, only where tumor cells are. So using a system called luciferase. So luciferase is the enzyme that's used by fireflies and fire beetles and other glowing insects. And so what they did was they took the gene for that, and I actually used this gene in mice for circadian rhythms, because you can actually plot it. You can look at the brain will glow during the daytime and then not glow during the nighttime. It uses luciferin as the substrate, and so it breaks luciferin and releases a photon, which is how they glow in the dark. And so you can actually look at that. And so this is tumor burden afterwards, and the tumors that are the ones that are mock, and PBS is just saline. That's just a phosphate buffered saline. So the animals that had been injected with cancerous cells so if you look at the top row, at day 14, the PBS and the mock mice are a little glowy compared to the 19BBZ. By day 21, the PBS and the mock mice are completely tumorigenic. They have tons and tons of cancer. And so much so that they never make it to the 1 month or the day 39 part of the experiment. They die. They all die. That when something is all, that's a pretty profound result. But if you look at the 19BBZ, for some reason, which we think we know what it is, based on this paper, they're able to fight off the cancer a lot more effectively, so more of them live longer.
So the brighter the color, the worse the cancer.
Right.
Is in that area.
Right. Correct. Okay.
X's mean that they're dead, right?
The X's mean they all died, yeah. And that's really short, because like mouse lives are short, but they're usually like left to their own devices, maybe 2 years. 2 years is not 39 days. So this is a pretty aggressive cancer that they injected them with. Okay, go ahead, Chin.
Okay, so for figures D, E, and F, it's just showing that the mice that were treated with 19BBZ cells had a decreased frequency of those tumor cells in the spleen, the blood, and the bone marrow, which kind of just points to the effectiveness of 19BBZ. And in the last figure G, it just shows that for the mice that were treated with 19BBZ CAR positive T cells, they all survived, which when compared with the others, with PBS and the mock groups. So that kind of just shows how effective the 19 BBC is. And here are the conclusions. So the non-viral nature of the Sleeping Beauty system, which is plasmid-based and can be delivered through the use of large volume electroporation, would reduce costs and the timeline within CAR T cell therapy. And the overall procedure is fully adaptable to closed systems for gene modification and cell expansion. And the adoption of this optimized manufacturing protocol would potentially pave the way for a broader use of CAR T cell therapy. And that's it.
Good. So, Chin, should this have been published in Gene Therapy?
Yes, in gene therapy. Oh, sorry. Maybe not for like other bigger journals.
Yeah. So I was telling Chin, I'm a member of the American Society for Human Genetics, and I review papers for their journal, which is called the American Journal of Human Genetics, which has a higher impact factor than gene therapy. It's one of the main journals that you would send a paper to if you're a really good geneticist trying to get a high profile paper. Even still, like, it's not one of the sexy journals. Like, sexy journals, Nature, Cell, Science, eLife, they have really, really high impact factors, whereas American Journal of Human Genetics is probably around, so that, like, theirs is 30-ish. American Journal of Human Genetics maybe more like 11. This one is more like 4. That's still respectable. That's a pretty decent journal, but I would have rejected this multiple times over if they had sent it to American Journal of Human Genetics, because there were some things in it that were pretty egregious, even in addition to what I already pointed out with the statistics, right? So if you go to figure, figure 5C, they were quantifying GFP. You can't, I mean, you could, if you're counting cells, that's one thing, GFP positive cells. But if you're actually looking at the amount of fluorescence, that's you can't do that. It's not reliable. So that I would have said something about that. They did this weird thing. Figure 2's weird. So if you're looking at the paper, you look at figure 2, and it is a stacked bar plot with error bars, which I don't think I've ever seen that before. Because normally this is what that would look like. The one stack of bars would actually be spread out over, there's one, and there's one. And then you give a space and draw the next ones. But I guess they didn't want to do that. I don't know why. I mean, they all add up to 100, but why, if it's percent, why are there error bars for percent? That's new. I don't know. I don't know what the answer is to that. Otherwise, though, like, if they'd run the stats right, I think they still would have ended up with the same result. So, like, what's the implication for real life, Jen?
Oh, for like the experiment?
For like, why should we care?
Oh, because it can help, like, it can help the efficiency of CAR T cell therapy, which in turn would help the efficiency of getting rid of, like, those B cell leukemia, lymphoma type cancers.
And the efficiency you're talking about really is making more CAR T cells in shorter amounts of time, right? Yeah. Because, like, that was the first part of the paper. How many can we make? And then the second part of the paper was, are they effective? We already knew they were effective. There is a CAR T cell therapy, and it just takes a long time and it's expensive to do. But now if we could do it faster and cheaper, right? I think that's where they were going with this. So, yeah. Okay. Do any of you guys have comments or questions?
How would you do that experiment faster and cheaper?
How would who? No, I think that was the point of the paper, is what I'm saying. The paper did it faster and cheaper than is normally done.
Yeah.
Yeah. Any other questions, comments, concerns?
Do you happen to know if there's been any further advancements since this experiment was done in like 2019?
I not to my knowledge. I don't think there's anything else. So I'm not sure.
Well, there was the original paper that you proposed, right? Which was this had been,
but that was based off of
right. That was based on this, right? So
Oh.
kind of giving an update on like where's the field going. So I don't think there were any. I don't think he mentioned any new findings, but that this was a big deal that hadn't gotten enough attention.
Thank you.
I have a question.
Sure.
So in the figure, like F on that one in figure C, like figure C, my bad, with the 19 dBz like column.
Which figure?
So this one.
C is the panel. Which? Oh, that one. Oh, okay. So figure 6.
Yeah. So at the end, like in the day 39 row, like with the 19 dBz, like rats or mice, the 3 of them are like filled with cancer, and then 2 of them don't have a speck of cancer on them. Why is that? Is it like maybe like something like a part of their gene, like makeup, genetic makeup?
I'm not really sure, but it kind of, I mean, compared to the others, it's kind of just a lot more effective. Even though it's showing that cancer in the mice, compared to the other 2, it's still a lot more effective, I guess.
So remember today, those UNAP, today we talked about drugs, and the effectiveness of drugs and the addiction and all that stuff really has to do with what type of receptors you make and how many you make, right? That's going to really affect it. So genetic differences could play a role if we're talking about you and me. However, these are inbred mice. They are clones of each other. They have exactly the same genes. So it can't be that. That's been controlled for. What it doesn't control for is just where the cells are injected when they're given the cancer. So usually they're injected IP, which means intraperitoneal. So they inject it right just here, right under the rib cage. Where you stick the needle is kind of haphazard. So if you hit a major organ or an organ that's protected from immune cells, you're going to develop bigger tumors than those that went into your bloodstream or something like that. So that might account for this, the differences between those mice. I mean, if you look at in the day 28 row, there are 5 mice. Look at the far right mouse, and then look at that same mouse on the next one. His tumors went away. So this therapy was actually reversing his cancer. Or at least, well, we don't know that for sure, right? We just think that might be happening.
Right.
Yeah. Okay. Anybody else? Nobody? Nothing? You got nothing? Okay. Well, thank you, Chin. That was very good. And I just want to point out, Chin made a concerted effort to make sure that she told us what the axes were actually telling us, right? The x-axis is this, the y-axis is this. You should be looking at that bar because that's going to show the differences. That's good. Okay, now the next step really is for someone like Chin to go, I just gave the background. Now one of you present figure 1, because that's how a journal club actually goes, right? She provides the background. She's the host, essentially, and leads the discussion. I've been doing that. I've been leading the discussion, but you guys who are presenting should be getting other people to do figures. Even if you have to arrange with them ahead of time, be like, hey, would you mind doing figure 1 later tonight? Yeah, no problem. Help each other out. That's what it's supposed to be, a group effort. Okay. So that was very good. The next one is Caitlin DeCrane. That'll be on the 29th. So next Monday. And she is doing, and this is already up, Martorell et al. 2019, multisensory gamma stimulation ameliorates Alzheimer's associated pathology and improves cognition. This is published in the hallowed tome that is Cell. Okay. Probably the best journal in the world. Of the big four, this is the best one. And so they tend to be really long and really detailed. So Caitlin will go through and prune out what's probably not necessary to go over, but it's already up and ready for you guys to read. Okay. And it's about Alzheimer's. I don't know if you have Alzheimer's in your family. I do. This will be interesting to me. Okay. 7:44. Well done, team. Okay. You guys are so excited. All right. Well, have a good night.
Have a good one.
Yeah, bye. Good job, John.
Thank you.
All right.
Thank you.
See you. That was a chart.
See you later.